Hello everyone, my name is Ayesha Maqbool and I am working as an assistant professor in the Department of Molecular Biology, Virtual University of Pakistan. In the beginning of my talk, I would like to thank the organizers for giving me the chance to participate in 17th International Conference on Pathology and Cancer Epidemiology. Today I have the honor to present the main findings of my research project that I performed in Adnan Menderes University, Turkey. And this is about the effect of in vitro fertilization and embryo culture on the expression of brain-derived neurotrophic factor, that is BDNF, mainly in the hippocampus of mice. So all of us are familiar with the social and psychological effects of infertility that is affecting around 7% of whole of the world population. And since the birth of first ever human baby who was conceived in a petri dish, she is Louise Brown and was born on 25th July 1978. Since her birth, a new era has started. Because after she was born, there has been around 4 million births using the assisted reproductive techniques. And fortunately enough, depending upon the requirements of the infertile couples, there are several different kinds of assisted reproductive techniques emerged so far, and these include in vitro fertilization or IVF, intracytoplasmic sperm injection, also known as ICSI, and in vitro embryo culture, the names of which I believe are self-explanatory. And in fact, what happens under these in vitro conditions is that the blastocyst, it is now exposed to such conditions which actually exclude all of the natural microenvironment that would be provided in the uterus. And as a consequence of this developing embryo is now exposed to several stress factors. And in turn, this situation can reduce the development of the inner cell mass or the trophoectoderm. And what can be the consequence of this? This can lead to the abnormal development of the fetus, or it can bring any unwanted changes in the embryo. And actually this is happening, that the children who are born with the artificial reproductive techniques, they have been reported to be at the higher risk for the certain conditions, like they have low birth weight, they have got premature birth, there is more tendency of admissions in the hospitals, and even the rare diseases like the epigenetic diseases. And these include Beckwith-Wiedemann syndrome, Prader syndrome, Engelmann syndrome, and even cerebral palsy and epilepsy. And what we have to notice here is that all of these clinical conditions manifest the alteration in neurodevelopmental processes. And along with this, some of the studies have noticed relatively low IQ levels in the children who are born with the artificial reproductive techniques, and even higher rates of autism spectrum disorders in these children, which are born through ARTs as compared to those who were conceived through the natural matings. And these are all the points to get worried about these children. So after these indications, the evidence was required which could demonstrate the link between the reproductive procedures, between these artificial reproductive procedures and the neuronal disorders. So for this purpose, several animal models were employed. For example, the model which was employed by Ecker and his colleagues. In that, he exposed the blastocysts to in vitro embryo culture and embryo transfer, meaning the embryos were transferred to the surrogate mothers. And when these mice reached the adult age, they were exposed to the behavior experiments in which they indicated the decrease in anxiety levels and impairment in the spatial memory. So the experiments which were employed are Morris water maze and elevated plus maze task. So Morris water maze is the task which is utilized to analyze the spatial memory. The animals are allowed to swim in the circular pool which is surrounded by the visual cues. An overhead camera records the activity of the animal. There is a hidden platform which the animals have to detect by learning its position over several days. And after the interval of training, the probe tests are performed in which the platform is removed and the time which they spent in this particular quarter is calculated as the measure of spatial memory. Whereas the elevated plus maze task is a plus-shaped paradigm in which there are two platforms with the closed walls and two platforms with the open walls. And the animal which spends more time on the open walls indicates a reduction or the decrease in the anxiety levels. So coming back to the other studies, the behavior deficits which were observed by Acker and his team members, they were also observed in another study by Fernandez Gonzalez when he exposed the animals produced by ICSI and some other animals which were derived from blastomere biopsy and in vitro culture experiments. They also exhibited some behavior alterations along with the reduction in the depression-like behavior and also the changes in their body weight. And all of these studies imply a clear connection between the use of artificial reproductive techniques and the clinical conditions which were observed in the individuals produced by them. So, therefore, we became interested to find the underlying reason for the deficits that were observed in these individuals. And for this purpose, we thought that the neurotropins, that are the factors which are known to perform several functions in the brain, they might be playing the observed behavior and clinical conditions among these animals. And among the neurotropins, which actually include neurotropin 3, neurotropin 4, and nerve growth factor, BDNF, which is brain-derived neurotropic factor, it has emerged as the key molecule and it has been found to play a role in the development of the neurons, in the survival, in the synaptic plasticity, and even in learning and memory. And in the studies, with the lack of function of BDNF, certain situations like neuroinflammation, neurodegenerative diseases, and even with the lack of BDNF, the apoptotic cell death was observed in the neurons. Therefore, we raised the question that whether the expression of BDNF is altered in the brain of mice which are produced by embryo culture or not. And for this purpose, we designed the following study. So what we did was that we cultured the blastocysts under the laboratory conditions, and after a certain time, these blastocysts were transferred to the pseudo-pregnant surrogate mothers by the surgical method, and these surrogate mothers were produced by crossing the C57 Black 6 and Barb C mice. And during this procedure, 5 embryos were injected in each horn of the surrogate mother, and when these mice were born, they were allowed to get adult and reared till the age of 6 months. After the age of 6 months, they were sacrificed and their brain parts were isolated for the analysis of BDNF expression. So along with this, a control group was also raised which included the pups which were born from the female mice in the estrus cycle, and they were mated with the male mice of the same age. So regarding the expression of BDNF, that was analyzed by employing real-time qPCR assay, we did not see a significant difference in the control and experimental mice. So we moved forward and analyzed the expression of BDNF protein by means of immunoblotting. And here you can see the expression of BDNF in the hippocampus of control and experimental mice. Beta-actin was used as the loading control. And here is the densitometric analysis. And we can see that although this analysis does not reveal a significant change in the expression levels of BDNF in either of these organs, in the hippocampus and cortex, but overall there is a tendency of reduction in both groups. And if we look at these plots individually, then we can see that some of the animals have a clear reduction in BDNF expression level. And maybe here I should mention that hippocampus is associated with the spatial learning and memory processes, and cortex is associated with several brain functions like thinking and decision making. So what we did next was that we analyzed the expression of BDNF by means of another assay that was immunohistochemistry. And here in this slide you can see the cross section of mouse brain, where this is the cortex and this C-shaped structure along with this conical structure is the hippocampus. And this structure is known as the dentate gyrus, which is shown here. And dentate gyrus is the region of adult neurogenesis and is particularly and exclusively associated with learning and memory. So this part is known as the upper blade of dentate gyrus, this is the lower blade, and this inner part is the dentate hilus. And here you can see that in the control mice there is a stronger expression of BDNF in the dentate hilus by the DAB staining, whereas in the experimental mice the expression is reduced in the dentate hilus. And here the nuclear counterstaining was performed using the Myers hematoxylin. So in several previous studies, along with my own study, we had found that the reduced levels of BDNF are associated with the loss of neurons in the dentate gyrus and in the CA1 region. Therefore, we performed the Nissl staining, which is done to analyze the morphology of the brain, but we found no difference among the dentate gyrus of the control and experimental mice. And along with this, even in the CA1 region, there was no difference in the thickness of both kinds of animals. And actually this goes with the other results, because although there is a tendency of reduction in BDNF levels, but the changes were not significant. But after this, we found some very interesting results when we analyzed the brain to body weight ratio. So brain to body weight ratio is considered as an estimate of IQ levels and the intelligence of animals. And a point is that the dolphins have got the highest brain to body weight ratio among the cetaceans. So it was calculated by dividing the brain weight by the body weight of the individual, and then it was multiplied by 100. And this is the method which was published by Cairo and his colleagues in 2011. So we can see here that the graph indicates that the mice that were produced by the in vitro embryo culture technique, they exhibit a reduced brain to body mass ratio as compared to the mice which were generated by the normal matings. And I have to mention something here, that all of these experiments were performed with the female mice only. There was no male mice in this study. And here with this result, we can assume that some parts of the brain other than the hippocampus and cortex are also affected, because hippocampus forms a very small part of the brain and is very lighter in weight as compared to the whole brain. And this indicates that in future studies, the other parts of brain should also be considered. So we can summarize this study by saying that the in vitro culture of the fertilized embryo at the blastocyst stage that were transferred to the surrogate mother did not reduce the number of neurons in the hippocampus of the brain. However, it showed some tendency of reduced expression of BDNF in the hippocampus, along with the reduced brain to body weight ratio and the loss of learning and memory. However, the mechanism underlying this process is not known at the moment. So, with this study, we can conclude that at the age of 6 months, Barb C. B67F2 mice, which were produced by the in vitro embryo culture, they do not exhibit the major morphological changes in the brain. And most importantly, the in vitro embryo culture and embryo transfer techniques do not appear to alter the expression of brain derived neurotropic factor either in the hippocampus or in the cortex. These data also strongly suggest that the behavior changes which were observed by Ecker et al., Sampino et al., and others, they are either specific to the strains that were used in their studies or they underlie different molecular mechanisms. And however, the significant reduction that was observed in the brain to body mass ratio of the mice which were produced by the in vitro culture is particularly very interesting for the further investigations. So in the end, I would like to pay my gratitude to Associate Professor Dr. Levent Karagenj for guiding me through the project and for his helpful suggestions. I would like to thank our PhD student Kuksal Duan for taking care of mice and my very dear friend Hibba Salim for her helpful scientific suggestions. I would not miss the chance to extend my thanks to all the faculty members in the Department of Histology and Embryology, Veterinary Faculty at Adnan Menderes University, Aden, and Tubi Tak for the financial support, and last but not the least, thank you for your attention. I miss the event and the participants of today's conference very much. Thank you.